Hello, I'd like to thank you for inviting me to the webinar series and giving me the chance to talk a little bit about the Meat Science and Animal Biologics Discovery Program. And I specifically want to focus today on food safety research prospects as a part of the Meat Science and Animal Biologics Program. Okay, we're going to start over. So thank you for inviting me here today to talk about a little bit about the Meat Science and Biologicals Discovery Program, which is a new program that's being started at the University of Wisconsin as part of the Department of Animal and Dairy Sciences. It's an evolution out of the former muscle biology group, and now it's expanded into other areas as well. What I specifically want to focus on today is the food safety research prospects, which is of another angle that we want to take within the program to go along with the meat processing and everything else that we're doing. So if we take an overview of the Meat Science Animal Biologics Discovery Program, it has several components to it. Some of the obvious ones, of course, is meat and poultry processing and the muscle biology work that goes with that. And what we're going to talk about today, food safety aspects of that as well. Some other parts of the program which are going to be fundamentally important as we move forward is animal biologics. And animal biologics includes non-meat uses of animal meat products. In other words, the idea is that other applications that aren't necessarily consumed as meat, such as medical applications. Think of such things as, for example, heart valves from swine that can be used for human surgery and those sorts of things. And so that's another component of the program to go along with the conventional processing and food safety. Obviously, there's teaching and extension outreach components as well, and these will complement the research part of the program. And the idea is that these all are interactive with each other, and and as we develop the research, we certainly want to teach and train the next generation of meat scientists and food microbiologists coming out of the program, and at the same time work with the public to demonstrate some of the practicalities of what we're doing. So let's take a step a little farther in and look at food safety particularly. And I think this gives a good diagram and aspect of general food animal microbial ecology. And as you might guess, microbes can come in contact with food production systems all the way through the entire food system chain, all the way from the field for agronomy crops or the animals being grown on pasture and feedlots, etc. And so there's constant exposure to a whole myriad of different microbes. And these microbial populations obviously are going to be different at each aspect of the production system. I mean, live animal encounters with microbes is going to be different, for example, than say processing, what happens during processing and the microbes that are present in the processing plant. And so we are constantly in concerns of two things. Number one, obviously, is pathogens, and pathogens can certainly exist at all aspects of the production system. Salmonella is a good example. Salmonella is a good surviving type organism that can survive a multitude of different environmental stresses, et cetera. And so it's not unlikely that animals during growth will encounter salmonella, and certainly it'll be prevalent. It certainly will be present in the processing plant as well. Given that, it's no surprise that there's been a lot of focus on what we call combinations of different ways to either eliminate or decrease contamination. And obviously with pre-harvest, we have such things as antimicrobials that can be fed, vaccines that can be administered to the animals to help them mount an immune response against specific pathogens, and such things as pre and probiotics, which are more supportive of what we call the good microbial gut organisms, which then can serve as a barrier for pathogen colonization. For processing, we have a variety of approaches that present hurdles for pathogens to be able to diminish their growth and or eliminate, including antimicrobials. There's a whole spectrum of chemical antimicrobials, parasitic acid, among others, that are commonly used in the industry now. Temperature, of course, is another aspect. Using chilling during poultry processing, for example, helps to limit microbial growth. And then, obviously there's certain things that can be done at transport, consumer information and education, training of food handlers and food systems and food retail operations. All of those contribute and are important parts of the component. Now, the other thing that we have to consider about microbial ecology, it isn't just about the pathogens. It's also about the non-pathogen populations. And where we get concerned about that is especially in processing, is the whole business of organisms being on carcasses and leading to shorter shelf life, spoilage, et cetera. And so part of our intervention strategy is not just to eliminate pathogens, but also limit the spoilage organisms that may cause us sensory problems with certain meat products and those sorts of things. So all of that fits into this, and it's a combined interactive effort in terms of trying to develop as many hurdles and education processes as we can to prevent and limit the incidence of foodborne pathogen disease, as well as maintain freshness of the food product. Now, the two big areas, and I've obviously listed them in details, that we're going to focus on today is pre-harvest and post-harvest, and some of the strategies for those particular research areas. So let's start with pre-harvest food safety. And I usually like to combine pre-harvest food safety along with gut health, because to some extent, they go hand in hand. And the way we approach it is we've developed what we call a high throughput gut in vitro screening system. This is mostly based off of poultry, which we focused on a lot when I was still at University of Arkansas for obvious reasons. And then follow up with in vivo challenge studies, and both involve challenging with a particular pathogen, whether it be Salmonella or Campylobacter, and then bringing in some kind of feed additive, a prebiotic or a probiotic or an organic acid or whatever, and asking the question, under these types of matrices of gut microbiota, do we get inhibition or limiting of growth of a particular pathogen? In other words, is there indications that we can lower the levels of the pathogen under these circumstances? And as a part of that, we don't just look at pathogen numbers. One of the tools that's come into play that's become more and more popular and provides a considerable amount of information is also at the same time using microbiome 16S gene targeted sequencing to get an idea of what the entire microbial community is doing. And this is important because what we worry about a lot with a lot of feed additives is that sure, it will reduce the pathogen loads, but what you don't want to do is you want to basically do no harm to what we would consider beneficial gut microbes. In other words, there are certain gut bacteria such as Bifidobacterium and Lactobacillus, which are actually contributors to gut health and contributors potentially to animal performance. And you certainly don't want to introduce a feed additive such as an organic acid, which would actually also diminish those organisms as well and basically counteract whatever positive gains you have by limiting the pathogens. So when we generate this kind of data based off of sequencing, it allows us to do several things. Identification of taxonomic identification, what we call representative gut microbes. We get the entire microbial population, but what we're interested in is actually what we call a core or signature microbiome that is indicative of the presence of a particular feed additive, such as a prebiotic. For example, certain prebiotics are very selective towards Lactobacillus and Bifidobacterium, which are, as I said, a couple of beneficial bacteria. Depending on how consistent that response is, we would start to consider them core signature organisms that are associated with the presence of the feed additive, which has important practical consequences because that means that we can say the prebiotic is working because we see certain microbes increasing in their proportionality in the gut because of the presence of that feed additive. In other words, it's definitely a cause and effect, and that's a very important aspect from a practical standpoint. I mean, is your feed additive working, essentially, is what you're asking. We also can do what we call diversity comparisons. Obviously, you want to know if you have treatment differences. In other words, if you include the feed additive, okay, you get the signature population, but is it a detectable difference between that and, say, a control that doesn't receive the feed additive? Again, this gets back to the cause and effect. If we can attribute a certain microbial population shift in the gut to the particular feed additive, then we know that the feed additive has a good chance of being a positive contributor to overall gut health and limiting pathogens. Now, as we get farther into this, we're realizing that the gut system, we have realized this certainly, is that the gut system is a quite dynamic system. And so lots of times you may not change the composition of the gut microbiota, but you may change their metabolism. That's not uncommon, and it's not a surprise as we understand more about gut microbe fermentation, et cetera. So metabolomics, where we actually go in and identify all the metabolites that are being produced by these microbial populations, as well as host gut responses. I mean, how does the gut respond to these changes? And transcriptomics of some of the tissue level, say, amino acid transporters, et cetera, are those being affected? And can we relate that to animal performance? Immunology, immune response. The immune system is very interactive with the gut microbe, and knowing that interaction can impact how we want to approach our feed additive strategies. Proteomics, certainly in terms of proteins present in the gut. Where we want to go at MSABD is eventually what we call longitudinal studies, combining what we see with live animals and the responses, and then what does the meat quality and meat microbiology look like of the animals that are slaughtered after receiving these types of treatments, and is there connections between the two? And that's something that's never been really looked at very much, and certainly the MSABD with the BSL2 facilities, et cetera, for slaughtering, we can absolutely get at that question. And I think going forward, that's going to be important for the food animal industry. So what are some, what I would call potential user or useful outcomes or potential outcomes that would have practical aspects for the industry? And I think we've alluded to this some already, but certainly standardization of feed additives in terms of how we evaluate them. We've had several years of experience working with feed additives, and I can tell you that, there still is a lot of anecdotal data out there in terms of positive benefits, et cetera. And we're hoping with, more precise gut microbiome composition analysis and metabolome analysis go with that, to where we have both who's there and what the functionality of those organisms are that are there, that we can actually start to get what we call quantifiable data that we can start to do some measuring in terms of actual impact and potential effect. Obviously, pathogen reduction in animal performance, how consistent is that? Is that a predictable is that a predictable indicator for a particular feed additive? Again, the industry needs more of that kind of preciseness in terms of being able to evaluate whether they want to invest in using a particular feed additive. And as I've already mentioned, the longitudinal studies, we might not see that many effects on the farm, but if we consistently benefit at the processing side, for example, and we've seen this, if you increase white meat yield in poultry processing, well, that has value to the processor. And so even though you didn't maybe see animal effects, the fact that you've changed that composition of that meat product into something that has more value, still in the long run has value for the overall food animal system and the production and processing that goes with that. Prebiotic mechanisms and discovery is an obvious one. I mean, that's, as an academic, that's certainly what we're interested in. And what we're realizing is as prebiotics get a wider range of sources of prebiotics, et cetera, there's certainly some complexities with structure of prebiotics. I mean, cereal grain brands and other things have now been identified as having prebiotic properties. And we are currently working on trying to sort through which brands have the most benefit, et cetera. That also means a complex gut microbe response. And again, no surprise. I mean, the more complex the substrate is, the more complex the microbial population in the gut is going to be that responds to that complex substrate. And so, again, the microbiome data analysis and sophistication we have now, we can really start to get at that question in terms of, what the overall microbial population in the gut is doing in the presence of one of these sources of prebiotics. And so one of the things we're interested in doing is using the gut microbiome to identify active grain brand components. What we find is not all brands are equivalent in terms of response from a pathogen reduction standpoint, and the microbiome also tends to be different, the gut microbiome. And so we think that that is probably a reflection of differences in composition of the brand. Could be very subtle differences, but it may be enough to trigger signature microbial responses that are identifiable to a particular brand cereal. We've done a lot of work with rice brand cultivars, for example, and we've seen some cultivars be very inhibitory towards salmonella and others not inhibitory at all, even though you would suspect they'd be similar in composition. And so I think we have an intriguing tool now that we can start to use to start screening some of these candidates from cereal grains and ask the question, okay, which ones of these are more likely to be a consistent effect when we feed them to animals. And one side note is we're also getting into the genetics of the pathogens themselves and asking the question, how is the genetics of the pathogen interacting with the gut microbiome and therefore interacting with the feed additive? What genetic systems are affected, and can we explain reduction of pathogens by impact on particular genetic systems, and therefore we see the resulting phenotype of reduction of that pathogen under those certain circumstances. Again, the whole thing is much more complex than what we thought when we first got into this, but we now have some tools to start sorting out some of that complexity. And all of this, I think, will lead to commercialization and discovery opportunities. Both probiotic and prebiotic candidates, I think, will be easily identified based on these approaches. And so we very much anticipate being able to develop a whole portfolio of different organisms and different prebiotic compounds that may have some actual practical benefit to the industry for commercial use. So now let's go to meat processing itself. And strategy is a little bit similar, certainly from a tool standpoint. We again want to apply next generation sequencing, 16S microbiome sequencing, but the application is somewhat different than the live animals you might expect. One of the things we've done early on, and we're trying to continue to, and certainly in MSABD, we can do this even to a greater degree than we've ever done before, is the whole business of comparing conventional plating for getting at microbial populations on carcasses, and then comparing that with microbiome mapping of during the different meat processing steps. We've certainly done some of that for poultry already, and it does make a difference at which steps in meat and poultry processing, what the microbial populations look like. As you might expect, for example, the evisceration step is going to reveal a different population than, say, one of the later on steps in processing. And so that's been exciting because we're starting to get signature populations for different steps in processing. To go with that, we want to get into more, and we're working on pathogen quantitation. We really want to know absolute numbers of pathogens in conjunction with the mapping that we're doing from a microbiome standpoint. Again, obviously taxonomic identification of representative indicator microbes. Lots of times pathogen levels are very low in processing, and just trying to detect those and use those as processing controls is somewhat difficult. So the desire is to come up with indicator microbes which are much higher concentrations and much more consistent concentrations. And these organisms behave very similar as the pathogens do, and so we can use those to evaluate our interventions. In other words, if the indicator organism goes down, we would assume if any pathogen was present, it would go down as well. And with microbiome sequencing, we can actually develop signature microbial populations. In other words, more than one organism that might serve as more of an indicator microbial population rather than a single indicator organism. And again, I think as data gets more sophisticated, we can really use this as a really nice process control in terms of saying, yep, this particular population is present, behaves like the pathogen, and so it's gone down. That intervention should be successful. An interesting sidelight that we've gotten into is microbiome sequencing has actually helped us to do some screening and evaluation of non-selective and selective plating methods. In other words, we've been able to use that to validate plating methods, particularly the selective methods. Campylobacter is a good example. There's a number of selective media on the market for Campylobacter. We went in and literally microbiome sequenced pooled plates. In other words, we took all the colonies on a plate, combined them, and then ran the sequence on those plates. And what we found is that Campylobacter selective plates weren't as selective as they were being touted to be. And we would have situations where all the colonies on a plate, maybe only 5% were actually Campylobacter. The rest were something else. And so, and probably the even scarier component there was that was variable depending on the processing step. And so, again, we're becoming more and more convinced that that's a nice additional tool that one can use to evaluate, which selective media should we use? Well, you can use this as a way to say, okay, which is doing the best job of being selective, and more importantly, which one is consistently selective over the course of an entire processing procedure. And some potential outcomes that we see at MSABD for meat processing and food safety research is we look to continue to develop rapid quantitative methods for pathogens, especially for things like risk assessment. You really need to know how much of the pathogen is there, not whether it's there or not, but what is the actual level of the pathogen. And so, and ideally, you'd like to take it even farther than that. I mean, knowing total salmonella is helpful, but if you knew which serovars were there, that would be even more helpful, because certain ones are much more public health threats than others. Such things as CRISPR typing, DNA barcoding for tracking. There's some genetic techniques now in place that we can potentially do more rapid, not just quantitation, but actually serovar identification, et cetera. And I think going forward, those are going to be very powerful tools for getting a better handle on salmonella prevalence in processing, not just for poultry, but across all meat species. Quantitative microbiome mapping, knowing who's there is nice, but if you knew how much of each organism was there, that would be even better. And that would tie into being able to better quantify microbial loads, and not just know, like, say, for example, total plate counts, but actually knowing the composition of the total counts, and in terms of who's there. That would help us to develop predictors for spoilage, et cetera, because we would know not just particular organisms are present, but how many of those organisms are present, and are our processing methods selecting for particular spoilage organisms. Post-harvest microbial indicators, we've already talked about this some, certainly identifying the best indicator organisms to help optimize intervention performance and shelf life prediction would be very ideal and useful to the industry. Data analytics is something that's going to get to be more and more important, certainly with whole genome sequencing, which I'm sure you're familiar with. This microbiome mapping that I'm talking about, plugging these into risk assessment formulas. We have done some of this already, but we're going to need further collaboration with advanced computer programming experts to help us develop specific data set analytics that we can analyze this kind of data and be able to match up data sets from different sources of data generation. And then, as with anything else, I think commercialization is optimal here as well. I mean, certainly rapid pathogen ID quantitation kits are already being developed by certain companies, and we're in partnership with certain companies to develop these assays for precise spoilage prediction, which is a little bit farther down the road, but I think is also an important application to consider. So from there, let's talk a little bit, let's put our crystal ball on a little bit, and let's talk a little bit about current and future trends in food safety. Some of these have been around for a while, but they continue to grow. There's continued, for example, organic, natural, and the whole business of removing antibiotic growth promoters. That has really changed the livestock industry dramatically in a couple of different ways. Removal of antibiotic growth promoters has certainly driven the drive to look for alternatives to antibiotics. I mean, antibiotics had certain benefits. Coming up with ways to replace that to maximize gut health and animal performance is a huge driver in the industry right now. The organic and natural sources of meat products is continuing to grow. There's a lot of interest in this, and the procedures for growing these types of animals in these conditions can be somewhat challenging. And so I think we're being faced with, how do we keep those particular food products safe? And so we need to come up with a set of acceptable feed additives and acceptable antimicrobials that can be used in these types of process conditions. Alternative meats. Certainly, I'm sure you're very familiar with the emerging of plant-based meat products and culture-based, et cetera. That continues to grow, and drivers include animal welfare interest in terms of, coming up with non-animal types of sources of food products. Sustainability is something people maybe don't think about much, but sustainability is certainly in the picture as well. How environmentally demanding is animal production versus, say, comparable plant production. And I think it's important here with alternative meats is what we have to do is really be very clear in terms of how we're going to define meats in general, and where do alternative meats fit in that. What's the sensory qualities, what's the texture qualities, et cetera. And I think there's opportunities to do quite a bit of research in this area, and I don't think we should shy away from that at MSABD. And certainly the plan would be to definitely take a good hard look at what alternative meats potentially should look like and what they should taste like, et cetera. One of the things that's becoming more prevalent, certainly in some of the processing industries, is the labor shortage. There's a big interest in going more and more towards automation and processing, and there's several drivers for this. Just scarcity of labor in general has certainly become a factor, but COVID has accelerated that to some extent. And so there is active research in automation, and certainly we should be in a position to anticipate fully automated processing in the, coming in the future. And I do think that we are in a position in MSABD to be a partner in terms of looking at what that might look at versus conventional manual type of oriented processing, and how would we work in such things as rapid methods. Could you tie in pathogen detection with automated processing and build in for sensors for particular pathogen? Could you do quantitation? Could you do microbial load assessments, those sorts of things? And can you tie that in with automated processing? And I think those are some questions that we'll need to ask. Big versus small animal agriculture. This whole idea of retail vertical integration is occurring now in small animal agriculture. People are raising small flocks of chickens. They're taking them all the way out to retail. And I think we have to be aware of that and realize that that is a segment of our overall livestock industry, and how, again, do we apply food safety, et cetera, to that particular aspect of production. Big data revolution. Some of you, I'm sure, are very familiar with blockchain and its introduction by Walmart and other large retailers. Traceability is becoming important. I mean, being able to trace a meat product all the way back to the particular animal it came from is something that's gained a lot of traction. Cybersecurity. How do you protect this data? In terms of if you have all this data, who has access to it, and how can you prevent other people from having access to it? We've heard a lot of scare stories in the retail industry about cybersecurity, and this all goes with if we get into doing lots of sequencing, both whole genome sequencing as well as microbiome sequencing, we're probably going to want to protect that data. And so we're probably going to need some interaction with computer engineers to help us to do cyber protected type codes and things for data. So what are the opportunities for MSABD? I think science-based identification of novel feed additives, we've certainly talked about, and the accompanying mechanisms that go with that is a great opportunity. We're already working on that, and I think we're going to expand that quite a bit. I kind of throw this out there because I think we need to think this way. Defining the concept of meat microbial ecology, I think with microbiome sequencing, we can actually do that term makes some sense, because we actually will know complete microbial composition of a particular carcass or meat product. And I think that has some real value to it. Animal biologics, we haven't really talked much about that, but the whole idea of maximizing use of animal meat products for uses other than meats, I think is going to be a very important part of the MSABD program. So the search for unique biologicals from tissue samples of animals, et cetera. But let's not leave out the microbes. The microbes in the gut, I think, is also a source of biologicals, both, obviously things such as probiotics, but other things too, metabolites, et cetera, that are generated by the microbial population. We should not ignore those. And those have some real potential commercial applications as well. I do think that collaboration with computer engineers to develop more advanced sequencing bioinformatics is going to be something we're already looking into, and we've done some of that already, and intend to expand that. And I think ultimately what we want to do is generate groundbreaking, innovative, peer-reviewed research at MSABD, and research that makes us the leaders in the field, that we're the go-to place, that people want to do this kind of research and go to us to solve problems as well, that we have the scientific horsepower to be able to do that, and we certainly can anticipate some of the things that's going to be needs for the industry as we go forward. So with that, I'd just like to thank you, and feel free to email me if you have any questions on my presentation, or if you want to follow up on some of the things I've discussed in here. I would certainly welcome that. And once again, thank you for having me here, and I look forward to meeting everyone in person at some point when we get the chance.